Let's have a look at the mutations, folks, what they were. So DNA is normally a very stable molecule, okay? And so it's stable for reasons that we have already discussed. So firstly, there's a huge number of hydrogen bonds there, isn't there? Between every single pair of bases, there is a hydrogen bond. And so collectively, that means it's very strong. And because it's in a delta helix, okay, it's not immediately available to react with other chemicals. So it's very non-reactive and what you might call inert, okay? And that's a good thing. And of course, the other thing that happens to DNA, that normally it never leaves the nucleus. Okay, so it just sits there. And that also helps to keep it safe. I'm having a right devil to move this on. Oh, there we go. But as we've talked about, mutations can occur during DNA replication, either mitosis or meiosis. And also there are environmental factors that are going to increase genetic mutation. Okay. So let's remind ourselves what a genetic mutation is. So genetic mutation is a change in the sequence of coding DNA, okay, leading to often a change in the protein, okay? And if you change the protein, you may change the ability to bind, okay, to form hydrogen bonds. If you change the ability to form hydrogen bonds, you may change the shape. If you change the shape of the protein, it will not function. It will either become dysfunctional or it will function in a different way. Okay? So, when we're talking about mutations, folks, we're talking about substitution, deletion, and insertion. Okay? And so these are fairly obvious to understand what they mean. So look at this first one. We have substituted G for A. This is the original strand, CGC, GUU. So the substitution has occurred here. These codons obviously code for an amino acid. Arginine and serine are unaffected, but allene here has replaced valine. So that's a substitution mutation, and only one amino acid is altered. The rest of the protein is okay. And so that's quite a subtle mutation, and one that might have an evolutionary use if we're trying to get a lot more variety without too many deleterious effects. In the deletion mutation here, you can see the G has gone altogether. And what that leads to what is known as a frame shift. And that means everything after this deletion becomes a different amino acid. So we've got, whereas we had arginine, valine, serine, we've got arginine, phenylalanine, proline. Okay, and so everything beyond this point is changed. In large organisms, that's likely to be pretty devastating, a devastating mutation for that organism, okay? But I suppose it depends at what point in the gene, that if that occurs towards the end of the gene, then only the amino acids at the end of the gene will be affected. If that occurs at the start of the gene, you would get radical change. Now, whether that would be the mechanism, you would say that's normally the mechanism for absolute disaster in evolutionary terms. I suppose by random chance, you could end up with a whole completely different organism that way. It's very hard to see how it wouldn't just kill the organism or its offspring. We don't need to go into that. And it's the same thing with insertion here. Okay, so we've added an A here. We've had a frame shift. Everything beyond, every amino acid that we add beyond the A we get a totally different outcome. Now, as well as these types, already substitution, they could have no effect. And the reason they may have no effect is that the code is called degenerate. Sometimes you might change a base there and you still would get the same amino acid coded, okay? Because it's called a degenerate code, meaning more than one sequence leads to an amino acid. You can have what's called a missense mutation, a single amino acid change as stated. If you end up with a stop code on there, that's called a nonsense mutation. And then obviously the protein doesn't get fully built, it's only half a protein. So deletion mutations is a frame shift. All amino acids downstream are affected. You've got a completely non-functional protein there most of the time. So another type of mutation that does occur is problems in mitosis or meiosis with the process itself. So let's look at this one. This is Down syndrome here. So looking at this briefly, we've got something called non-disjunction occurring in meiosis 1. Okay, so let's remind ourselves of meiosis 1. What should happen is in meiosis 1, the homologous chromosomes should line up, okay, and there's the centromeres, and the spindle fibers should attach to the centromeres. Those spindle fibers contract, and the whole chromosome goes to the other side. Now the question is, what if in this case the spindle fibers do not connect with the centromere? Okay, this contracts, there's no pull of the chromosome to this side. We can end up with one side having 24 chromosomes, okay, and the other side would end up with 22 chromosomes, okay. These 24 chromosomes end up in the sperm or egg. Also in this case, the egg is shown as the one with the extra chromosome there, okay? And so this sperm still has 23. When we get together there, there's going to be 47 chromosomes. The offspring is viable. The offspring should be sterile and will also have Down syndrome, okay, features. Now, the prognosis for Down syndrome has got better and varying functions, but basically they will function as a child for most of their life, at the level of a child for most of their life. I think it's fair to say. Okay, so that's one problem. On the year 13 course, you'll also look at inversions in chromosomes. It's not on the year 12 syllabus. On the year inversions in chromosomes. So other problems in chromosomes can occur to feed into this topic. Okay, it's time for you to do a little bit of work before I go on to cancer. As I go on to cancer, really, cancer is mainly studied in year 13. So although I build this as a revision lesson for year 12, which it was with all the DNA replication and the mutation stuff is on the AS course, but I just think you can't really interestingly talk about mutation without talking about cancer. So we will go into cancer today, although that, as I say, this was billed as year 12 revision now, but there is some new learning for you here today that will have to be covered again in year 13, I think.